Hi everyone, my name is Ron. I am a postdoc in John Glass's group, and today I'm going to tell you about some of the work that I've been doing on the JCVI minimal cell. First, I'm going to tell you about an experiment that we've been trying to develop in collaboration with Zan's group at the University of Illinois and TJ's group at Harvard in terms of trying to pinpoint and visualize the origin of DNA replication in the minimal cell. I'm going to tell you a little bit about Hub A, which is a histone protein, part of a larger family of nucleoid associated proteins. We heard Roseanne talk about that earlier and how we think that is influencing chromatin structure and organization in our minimal cell. And then unrelated, but re related to what Zumra talked about in the previous presentation, trying to understand exactly how whole genome transplantation works and some experiments that we're trying to establish to try and figure that out. So first, I'm going to start with the DNA replication stuff. So these are some micrographs that were generated by Ji Wang, a graduate student in TJ's lab, looking at fluorescently labeled FTSZ. So this is in JCVI Syn3B, which has a landing pad for easy installation of genes at a non-native locus. FTSZ, we heard Alejandro talk about FTSZ, forms a ring at the center of a cell when it's dividing. And this is a very interesting pattern because in natural bacteria, if you look at fluorescently labeled FTSZ, what you see is a layer of fluorescence in the middle of the cell. What we see here with the JCVI minimal cell is fluorescence not only in the middle of the cell, but all around the cell, suggesting that the cell may be dividing at multiple spontaneous random points inside the cell. So this begs the question, where is the origin of DNA replication and what path is the origin of replication taken? So for a long time now, we've been trying to develop an experiment where we create a Syn3B cell that has an additional copy of two genes, one a fluorescent label on FTSZ and another one a fluorescent label on DNAA. DNAA is a protein that binds to the origin of replication. It's a universally conserved protein that binds to the origin of replication and initiates DNA replication using a combination of AlphaFold and published literature based on what's been done in E. coli. I placed the mCherry label in the middle of DNAA. With the native DNAA promoter and with other strong promoters, unfortunately, this proved to be difficult because it seems like the cell is unable to handle an additional copy of DNAA if you put a label on it. We were able to get viable Syn3B cells if we put a weak promoter onto DNAA, but when we did that, we wasn't able to get fluorescence. So we had to come up with a different way of trying to pinpoint the origin of replication in the minimal cell. So Nika came up with this idea of trying to visualize the origin of replication in a slightly different way based on a methodology that was developed for looking at DNA in mammalian cells. This takes advantage of fluorescently labeled dCas9. So wild-type Cas9, based on a user-defined single guide RNA sequence, will bind to a specific DNA sequence and introduce a double-strand DNA cut at that sequence. dCas9 is a mutated version of Cas9, also known as deactivated Cas9, that
Still in the workshop,
will
but that's
bind
okay.
to a DNA sequence based
It's
on a
not.
user-defined single guide RNA
But
sequence. But instead of introducing a double-strand cut, what happens is that the dCas9 protein is just going to sit there. So in mammalian cells, if you put a fluorescent label on dCas9, you can just visualize fluorescence at that specific spot. If you add some additional elements to such a DNA sequence, specifically the MCP protein, MCP is short for the MS2 coat protein. This is a protein that is naturally present in bacteriophage MS2 that infects E. coli. That protein is known to bind to a RNA sequence known as the MS2 sequence. If you attach a different fluorescent label onto that and have, 15 to 20 repeats of this MS2 sequence, wherever the dCas9 goes, you can really blow up that fluorescence signal. So what we want to do is adapt this methodology and target a non-essential gene that is relatively close to the origin of replication, as you can see here. And because TJ's lab has access to really high resolution microscopes, the advantage with DNA-A is that DNA-A labeling allows us to look directly at the origin of replication. If we take this methodology and insert a big piece of DNA at the 0907 locus, which is roughly 4 kb away from the ORI, whether we look directly at the ORI or approximately 4 kb with the ORI, from the ORI might not make that much of a difference because of the type of microscopy that TJ is able to do. So right now I have a piece of DNA that has all of these sequences with different fluorescent labels, but adapted for our purposes, specifically containing mycoplasma regulatory sequences. And we're hoping to transplant this pretty soon, and hopefully we can send some cells to TJ so that we can look at the origin of replication. Next, I'm going to tell you about HUP-A, which is this histone protein that I've been working on for quite some time now. So one way of looking at how chromatin is organized in the cell is using a chromosome conformation capture assay. This is an experiment where you cross-link DNA in living cells with something like formaldehyde. You can separate cross-link DNA with non-cross-link DNA with a restriction digest. And then what you can do is that you can sequence those fragments and create what's called the contact map of the resulting data. So in E. coli, if you look at this two-dimensional contact map, this diagonal line essentially represents the chromosome. And what this is showing is that E. coli is capable of exhibiting a lot of interactions all along the chromosome. When this experiment was done with SYN3A, this was very surprising. It looks like there's no point of contact along any point along the chromosome. And this is interesting because we have a cell that expresses every single gene, but may not have a defined chromatin structure. And we think this is very interesting from an evolutionary standpoint because we think that by studying chromatin organization in the minimal cell, we may be able to get a glimpse of what primordial cells looked like early in the evolution of life before cells develop crazy sophisticated ways for organizing their chromatin. So if you look at the genome of SYN 1.0, upstream of HUP-A, this histone protein, is GPSA. Let me minimize this. In SYN3B, GPSA was removed during the minimization process. Proteomics data based on E. coli, which has about 30,000 HUP-A proteins in a living cell. SYN 1.0 has about 6,000. That number has been significantly reduced to about 28, as Roseanne mentioned earlier in her talk, in SYN3B. Using Cas9 and yeast genetics, I was able to add GPSA back into the native spot. Our hypothesis is that the loss of chromatin interactions that we didn't see with the 3C mapping experiment is because this non-essential GPSA gene was removed during the minimization process. It's also important to note that the 3C mapping experiment wasn't done with SYN 1.0, so that's something that we're going to have to do in the near future. So I was able to put this back in the genome of SYN3B in yeast, and then I gave these cells to Zumra, our transplantation postdoc, and she was able to put this into a capricolum cell and boot it up. What I did was I prepared RNA from SYN 1.0, SYN3B, and SYN3B that now has GPSA added back into its native locus. And I did qPCR to measure the expression levels of both GPSA and HUP-A to see if reinstallation of GPSA into its native spot would restore the expression levels of HUP-A. So this is the qPCR experiment that I did where we can clearly see restoration of GPSA expression But surprisingly, it looks like if you add GPSA back into the native spot, you get a little increase in HUP-A expression, but not to the level that we see in SYN 1.0. So we were curious as to whether anything from SYN 1.0 might have been removed during the minimization process that may allow us to determine what exactly allows for full restoration of HUP-A expression, because qPCR data suggests that you can put GPSA back, but clearly it's not the gene that was immediately upstream that is responsible for this phenotype. So what I did was I went back to SYN 1.0. So when SYN 1.0 was minimized, the genome was minimized in 8 separate overlapping fragments. From each individual fragment, a certain subset of non-essential genes were removed and then recombined with the remaining 7/8 of the SYN 1.0 DNA to create 8 different strains that have different genetic combinations. So to determine whether there were additional factors from SYN 1.0 that are not present in SYN 3B that may allow us to explain what that may allow us to find something else that's not GPSA that allows for full restoration of HUP-A expression. I did the same experiment as before with all of these different reduced genome design strains. I measured the expression levels of GPSA, which I'm showing here on the left. We see no expression of GPSA in SYN 3B, which is what we would expect. Segment 4 doesn't have GPSA in it, so we shouldn't see GPSA here either, but we do see GPSA expression in the rest of the reduced genome design strains. And one important thing to mention is that this y-axis is not a linear scale, it is a logarithmic scale, so there actually is a big difference in the expression levels of a lot of these genes. We also measured the expression levels of HUP-A in all 8 reduced strains. And what we're looking for are strains that show minimal to no HUP-A expression relative to SYN1. So here I show the expression data of HUP-A. From left to right, we see expression of HUP-A in SYN1, minimal to no expression of HUP-A in 3B, and then the GPSA transplant. And 2 of the 8 reduced genome design strains stood out to me the most. That is 1 and 7. The other ones showed intermediate level of HUP-A expression. So if you look at some of the genes that were removed in segments 1 and 7, a few things stand out. This RPIR transcriptional regulator is associated with sugar and phospholipid metabolism and other bacteria. GPSA is a gene product that is known to participate in phospholipid metabolism. If you look at some of the genes that were removed in segment 7, a few things stood out to me. A C5 specific DNA methylase that can influence the expression levels of a lot of different genes and the nature of chromatin in some ways. A gene associated with a toxin antitoxin system. This one, its cognate antitoxin, has DNA binding activity, and another transcriptional regulator that is associated with sugar metabolism. As an alternative approach, we took advantage of the D series collection of minimal strains. So this was another way of trying to identify what genes to keep and what genes to maintain during the minimization of SYN 1.0. So instead of taking several fragments and splitting it up into 8 parts, a lot of non-essential genes were removed from SYN1 in a sequential manner. So SYN 1.0 has 31 genes deleted compared to its wild type, the original wild type Mycoplasma mycoides strain. If you take away an additional 41 genes from SYN 1.0, you get what's called D1. And then D2, D3 has, you slowly increase the number of genes that you delete sequentially. And we have 22 of these strains. D22 has 255 genes deleted. So I took advantage of these strains that we have, and I did the same experiments as before, extracted RNA, did qPCR, And I did this with D22 because it has the largest gene deletion in the set. And the idea is similar to before. If we see minimal to no expression in D22, the regulator for HUP-A is probably one or multiple of those genes that were deleted in that 255 set. So on the left, what I'm showing you here is RT-qPCR data, where we measure the expression levels of GPSA and the same strains I showed you before. Now we have D22. GPSA is present in D22. This is what I would expect. And interestingly, similar to what we've seen before, where we see minimal expression in SYN3B and in SYN3B with GPSA, we see minimal to no expression in D22. And so this suggests that the regulator for HUP-A is probably one or multiple of those genes that were removed in that D22 strain. What I did was like cross-references data to the reduced genome design qPCR data, and one of the transcription factors, RPIR, is also absent in D22, so that's interesting. The two other ones, the C5 methylase and the toxin, are present in all of the D series strains. And then the other transcription regulator has been removed from D7, which I haven't tested yet, but I'm definitely going to do that. So this data, based on all of this data, we have accumulated a few candidates that we think are very interesting, and we're definitely going to follow up on this stuff. So I'm really excited about that. Lastly, I want to tell you about some experiments that we have going on to try and understand the mechanism of whole genome transplantation. So one hypothesis that we have for how cells are able to take up a whole genome is that when you treat recipient cells with PEG and calcium chloride, PEG serves as a crowding agent and is also a membrane perturbant. And we think that a lot of the cells are crowding around each other when you have genomic DNA around, and that somehow leads to a fusion event. So rather than an individual cell taking up a whole genome, multiple cells are fusing together to then take up a whole genome. This mechanism is similar to something that is known to happen in mammalian systems, known as cell syncytia. So in the context of certain infections with mammalian viruses, certain viruses will induce the formation of cells to come really close together, and all the cells come together in this one big blob, and this provides a big advantage for the virus, and we call this syncytium. There's previous data on Mycoplasma pulmonis and even in capricolum that suggests that cells may be fusing, at least under antibiotic treatment. So there's precedent for such a hypothesis. And what we want to do once we have an experiment that I'm going to describe to you in a little bit to try and explore this. In collaboration with Elizabeth at NIST, what we want to do is, under transplantation conditions, determine whether cells are fusing in a microfluidic setup. So to determine if cells are fusing, we are trying to develop a split aptamer assay. Aptamers, RNA aptamers, are non-coding RNA molecules that are capable of binding non-fluorescent ligands. These ligands are almost non-fluorescent, if anything, weakly fluorescent, but that fluorescence significantly increases when the ligand binds to the RNA aptamer. Broccoli is a commonly used aptamer. A lot of the aptamers are named after different types of fruits and vegetables, which is really interesting. This is the structure of the broccoli aptamer. What you can do is split the broccoli aptamer. So in E. coli, if you have cells that only make one half of the broccoli aptamer, you get no fluorescence. If you have cells that express the other half, you get no fluorescence. But in the presence of this non-fluorogenic substrate, and if you express both halves individually in E. coli, you get fluorescence. So we want to take such a system and adapt it to mycoplasma. P-myco1 is a replicative plasmid in both E. coli and in mycoplasma. The initial experimental setup was to clone these aptamer components on a P Michael 1 backbone and then put that into a capricolum cell, the recipient cell for genome transplantation. One issue that I had was cloning T7 on a P Michael 1 backbone. I could be wrong, but I suspect that cloning T7 RNA polymerase on a backbone that has a high copy E. coli vector is a bad idea. So I would do this in E. coli and I would get colonies, but T7 RNA polymerase would never show up on a plasmid sequence. To circumvent the issues of cloning T7 RNA polymerase on these plasmids, I took advantage of a T7 RNA polymerase expression system that Liji, a former postdoc in the lab, set up. So Liji created a landing pad system in SYN 1.0 and put T7 RNA polymerase in there under different promoters. Spirulin and 0602 are constitutive promoters, and she also put an IPTG inducible promoter in the system. What I did was, instead of making these P Michael 1 vectors with both T7 RNA polymerase and the broccoli aptamers, I just simply cloned all of the broccoli aptamers. I took these plasmids, I transformed them into Liji's strains, and then I selected for cells with the plasmid with chloramphenicol. I was able to do that. And once we get this assay up and running, the idea is that cells that express one half of the aptamer should not be fluorescent, or the other half by itself should not be fluorescent. But hopefully, under transplantation conditions and this non-fluorogenic DHF dye, we will get fluorescence in a microfluidic setup. So I'm currently working on this, and this is a first pass experiment with some transformants that I did. I think I'm gonna have to do a lot of optimization for trying to get this to work in mycoplasma, but in the negative controls, we don't see any fluorescence. And in cells that should be fluorescent, we didn't see that, but I'm hoping that it's a matter of just simply trying to optimize this for mycoplasma. And that's pretty much it. I want to thank everybody in our lab and all of our collaborators. And that's all I got.
Very nice, Ronald. Thank you. Questions for Ronald, please. Don't all speak at once.
Okay, it's me again. That's all very interesting, and this more like a comment or potentially, more collaboration. Daniel presented that last time, but we just tried a couple experiments with E. coli plasma, and they fuse so well. And we have two different plasmids that can be maintained in E. coli. I wonder if you would consider, if you are strains that we have for might be easy to always we did that and then to check that.
Bogomil, I lost you there for a little bit, but we should definitely set up a meeting and have a conversation about this.
Sure, yeah, because that would be maybe sometimes one system is slightly easier, but obviously you wanna have it, but for whatever reason, it could be beneficial. But
we would need your strains unless I've already got them, Bogomil.
Yeah, you don't have them, but unlikely, but still, that's not a problem. That's a couple days away.
Yes.
Okay, great job.